Hey everyone, welcome to lesson 6. I'm super jazzed about this lesson because it's about viruses. So, viruses and a little bit on prions, which honestly scare me a lot. So, we're going to be brief on that one. But viruses are very, very cool because they're in this interesting space where they're not quite dead, they're not quite alive, and they always have to be pathogenic. So they're what we refer to as acellular pathogens. They don't have the machinery to make cells, but they do have the machinery to hijack cells and force those cells to make more viruses. So completely unlike bacteria, fungi, or archaea, they are a lot of the times considered not alive. This is up for debate. There is a great virologist, Vincent Rocchianello, who has YouTube videos of all his virology classes available online. And when he first, was a virologist for a long time, he worked on polio, and that's what he did his career on. And he thought, oh, viruses aren't alive until I think it was around 2017. I saw an interview with him where he's kind of changed his mind to viruses are alive. They're just in this state that is similar to something like a spore. So spores, they can't, when they're in that spore form, replicate. But once they get the right conditions, they can replicate. And that's what viruses do. Once they enter into a cell and hijack that machinery, they start to replicate. So that's kind of why there's this interesting debate on are they alive? Are they not alive? So while they don't have cells, they do have genetic material. So there's this very clear delineation in viruses. It's one of the few things in biology that is very clear cut, where you have DNA viruses or you have RNA viruses. So, a quick overview is you have DNA, it gets transcribed into RNA, that RNA then gets turned into protein. So, it's where does the viral information enter into that system of producing protein is how we categorize viruses. Now, you might know about HIV and how that's retroviral, and that gets into some funkiness with exactly how that is categorized, but realistically, it's either going to be a DNA virus or it's going to be an RNA virus. And these are going to be wrapped in what we call a capsid. So that's a group of proteins that surround the viral DNA or RNA and also some viral proteins. So always you're going to have at least DNA or RNA, and you're going to have a protein capsid around the virus. Those are the two core things that all viruses will need. A lot of viruses package a lot of proteins with them, and those proteins aid in hijacking the cells and allowing the viruses to replicate. And of course, a lot of viruses have membranes as well because that helps with merging. You have a cell membrane, you have a viral membrane, and they merge together. And then that's another way that the viruses can more easily enter into cells is by using a membrane from ultimately taking it from the cell that they left last time. Now, they are obligate intracellular parasites. Now, I already said that they are acellular pathogens, so here's another definition for you. Obligate intracellular parasites, and that means they must enter a host cell to replicate. This means that viruses are actually pretty easy to prevent, whether it's your immune system or a treatment. One of the easiest ways to prevent viruses is stopping them from entering a cell. So if you can block cell entry from virus, then you're protected from that. So we have vaccines. The whole point of vaccines is so that we produce antibody and T cell responses to the virus. And part of that is producing antibodies that block viral entry. There's also, after a cell gets infected, there's a bunch of immunological things that happen to consume that cell, kill that cell, and move all that debris away from other cells. But the main way to prevent viruses from infecting you or infecting other cells after some of your cells are already infected is by blocking that viral entry via antibodies. Now, that is fairly easy to do because viruses are highly specific. They enter through different receptors. So a cell is a huge thing in viral terms, and it has a bunch of different receptors all dotted along it that let some proteins some in and not other proteins and do cell-to-cell communication. And viruses hijack usually just one of those modes of entry because they're very small. They don't have a lot that they can package with them in that capsid. So they're trying to get in as efficiently as possible and as quickly as possible. So them being highly specific is actually a boon when we are trying to keep them out of our cells. Now, there's a lot of different viruses. Like everything else in biology, there's literally a countless number. We have done experiments where we scoop up some water out of the ocean and try to do genomics to identify all of the viruses in there. And it's essentially never ending. The deeper you look, the more you end up finding. So people have published numbers, but every time they do, someone else comes back and revises the numbers and says, "Oh, there's a little more. Oh, there's a little less." So there's probably a certain amount that is the real range, but I don't think that we really know that right now because there's probably viruses with genetics that we don't identify because they're infecting weird things. And speaking of infecting weird things, the first category of viruses we're going to talk about is bacteriophages. So these are viruses that infect bacteria. Usually we think about viruses as, the cold, the flu, COVID, and they infect us, or something that infects animals. If you have cats and dogs at home, they have certain viruses that infect them. Also, zoonotic diseases, so diseases that infect animals, primarily livestock. Those we think about a lot, but we don't really think about viruses infecting bacteria, but that's exactly what bacteriophages do. So they have this dual cycle. One is lytic, where the virus hijacks the host and makes new phages inside of the host until the host cell bursts open and releases all of these now replicated bacteriophages. And then it has a second part of the cycle, which is called the lysogenic phase. So that is when the viral DNA integrates into the host chromosome as a prophage. So this is actually very devious on the bacteriophages side because it is not cycling, making a ton of itself, bursting out of the bacteria. It's actually integrating its DNA into the host. So that host will split and split and split and split and split and create a colony of thousands and thousands and thousands of bacteria. And at a certain point, they will then turn into the lytic stage where they burst open and they go everywhere. So they're hijacking not only the bacteria to replicate more bacteriophages, they are actually hijacking the bacteria's entire goal of replicating over and over and over and hijacking that process by storing their DNA in the host chromosome. And this gets at something with viruses in general, is a lot of them actually do this. A lot of them integrate DNA into chromosomes, and that happens in humans all the time. There's tons and tons of ancient, unknown viral DNA that is in human genomes and has been passed down over time. And the more we look into that portion of our DNA, it is usually termed dark DNA or the dark part of the chromosome because it doesn't have an active function that we can see. But the more we look at it, it's being seen not as something that is just useless junk, but actually has been co-opted by us to function as regulators for gene expression. So originally, someone got a virus that went in them, went into their DNA, and it's actually been kept through their entire lineage as something that the body then turned into a useful portion of its DNA. So there's some really interesting interactions, not just in the typical, you get an infection into a cell, the cell gets sick, the cell dies, releases more virus, but in the viruses come into cells, they deposit DNA into the chromosomes, and then sometimes that cell doesn't die, and the cell goes on and on and on. And this also gets into cancer. So there's a lot more data suggesting that cancer is caused by viruses. So there's human papillomavirus, which is known to cause cervical cancer, head and neck cancer, and there was a big study done with looking at people who got the HPV vaccine, so human papillomavirus, HPV, and they showed that all of the cancers caused by HPV did not pop up in that group that was part of this longitudinal 20-year study. They got vaccinated when they were kids, wasn't sure if it was going to prevent, 90% of cancers or 100% of cancers. That looks like it's preventing 100% of cancers by just blocking that virus from getting in and integrating into those cells. Similarly, Epstein-Barr virus, which is usually referred to as mono because it causes infectious mononucleosis, is implicated in cancer as well. It infects 90 plus percent of all adults in the entire world. And there are certain cancers that we have now linked to being caused by Epstein-Barr virus. And I think it is pretty easy to look at these cases and say, hey, there's plenty of viruses that we interact with on a daily basis that we're getting infected by on a daily basis. It would be crazy to me if we don't find more viruses that cause cancer and keep identifying, more and more precisely different viruses causing different subsets of cancer, especially. So that is something that's really, really interesting and why I wanted to lead with bacteriophages. But of course, you probably came into this thinking about animal viruses. So animal viruses are your typical virus. They infect us, they infect animals. We get sick, we cough on someone, they get sick, and that kind of just keeps going and going and going. Now, the way that these enter into our cells, whether they're DNA viruses or RNA viruses, is one of two different ways. It's endocytosis. So cytosis means consuming, essentially, and endo means into. So you're getting your cell membrane, again, has all those different receptors, and then a virus comes along, contacts the cell membrane, and the cell says, we're going to take you inside of us. You have all the right documentation to get into the cell. So we checked your passport, we gave you the stamp, welcome to France. All right, you're here. Uh, you know, they just are able to use that machinery to get inside. The other type is fusion. So fusion, instead of getting encased into a membrane and brought in, they actually have a membrane and the membranes fuse together and then it releases all the virus inside. So those are the two ways that they get into these cells. And there's also two ways they get out. One is lysis, similar to bacteriophages. There's just a buildup of so much virus that at some point the cell bursts and it releases all of these viruses out into the system. The other is by budding. So it's the opposite of endocytosis. Endocytosis means it's brought in and it's in a little vesicle of membrane. And then you have budding, which is just it's going to create a little vesicle and then release it out. And that's how you get membrane viruses. So some viruses, again, have capsids, and that's only protein. Some are membrane bound viruses, so they have a membrane shell and then a capsid inside that. Now, there are also, as I mentioned, retroviruses like HIV. They, instead of using DNA or RNA, use a reverse transcriptase to turn the RNA that they have, and this is why they're the ones that's a little confusing. Technically, it could be considered an RNA virus because that's all there's packaged into the HIV little genome. And it could be considered a DNA virus because it takes its RNA, turns it into DNA, and then the DNA integrates with our DNA. And this whole situation makes them, of course, the one thing that's a little bit odd and doesn't fall into those two really nice buckets of DNA viruses and RNA viruses. But we just put them in a third bucket called retroviruses, and that's how they do their infection. Speaking of infection, there's different types of viral infection. There's acute, which is your flu or COVID or cold, where you get the virus in you. It's a rapid onset, short duration. So you're sick for a couple days to a week. your body clears it out, you're feeling great again. There's also latent viruses. These are your human papillomaviruses, for instance. They get in and then they are latent. They're hidden, and every now and again they'll produce more virus. They'll spool up more virus. The virus comes out, infects you a little bit, infects those cells, hides its DNA into your cells, and then goes quiet again. And it's a latent phase. The same thing with chickenpox. So chickenpox can infect you, and if you get a chickenpox infection, hopefully you've had the chickenpox vaccine to avoid this, but if you've gotten infected with chickenpox, it can then in later life turn into shingles, which is just chickenpox essentially coming back. There's a different term for it medically, shingles, because it is such a severe disease and does not present the same way as chickenpox, but it is caused by the same virus. And then there, lastly, is chronic and persistent viruses. So these are ongoing chronic infections where you don't have that latent phase. There's always virus circulating. It's not hidden in your genome. It's always going to be wandering around. And this is HIV and the different types of hepatitis. So they're always circulating, always infecting new cells, always moving to new different cells, killing cells, budding off or lysing and going somewhere else. So when it comes to types of infection, those are the three main types of infection. Now, one of the things that has made viruses so difficult to study for a long time is they can't be grown with a normal lab media the same way you would grow, say, bacteria or even eukaryotic cells like HeLa cells. They have to be grown in living cells. So first you have to culture the bacteria, you have to culture the eukaryotic cell, whatever the host is, and then you infect it with the virus. And that's how you get the viral replication, and that allows you to study viruses. There's something called a plaque assay. So if you're looking at ways to prevent viruses, say from entering cells, you can put cells in a dish, and then you can treat those cells with something and then expose them to virus, and you can see if the virus still infects and kills off those cells. And the reason it's called plaques is because the virus will enter into one cell first and then kill that cell, kill more cells, kill more cells, kill more cells, kill more cells. And that creates these dots that you can actually pick out and see that we call plaques. So that's a really cool way of studying viruses and studying therapies to deal with viruses. Now, when it comes to diagnosing viruses, a lot of times it's very difficult because there's so many types of viruses that can cause similar symptoms. So when we're looking at respiratory viruses, for instance, I just got over being sick for a week with some sort of a lung head cold, and part of that is not knowing what virus it is, you just treat it the same way. You get rest, you drink a lot of Gatorade, you try and relax, you keep the fever under control, but all the symptoms are the same. Coughing, wheezing, you have stuffed up nose, runny nose, you get all of that throat runoff, you have a headache, you have a fever, you're dizzy. All of that is very similar, whether it's COVID, whether it's the flu, whether it's a cold virus, which are rhinoviruses, and there's a whole group of rhinoviruses, which is why we can't just vaccinate against the common cold or just treat the common cold as one thing. And then there's a lot of different things like RSV, respiratory syncytial virus, in kids and the elderly, it can be very deadly. But in someone who's my age or your age, you get it and you get it cold symptoms. And we just would call it a cold and not realize that we've been infected with those things. And this is compounded by the fact that, in the Southwest, I could get exposed to valley fever and it might present that same exact way, or a different fungus might present the same exact way. And I would never know because I'm just getting infected. I'm sick for a week and then I get better. So it makes it really difficult to diagnose whether it's a viral infection or not, and then also what virus it is. So there's a lot of different tests we have. Of course, there's uh, they're called LFIA tests. They're the ones where you swab your nose and you put in some, I'll call it extraction juice, and then you put it on the strip, and the strip has multiple lines depending on what type it is. One to tell you the test worked, one to tell you what you're infected with. Some strips have multiple lines for multiple different things. When I was sick, I took one that was flu and COVID to make sure I didn't have either of those. And we have those tests for at home, but to nail down exactly what virus it is, you have to do some sort of genetic testing. Usually that's going to be PCR. So that takes all the genes that you can get out of that sample, has different promoters to say, hey, this is, or sorry, I'll simplify it, has different tags to say, this is this virus, this is this virus, this is this virus. You run it through the sequencer, you see what actually comes out the other end, and then you can say what virus it is. But those are usually only being used in the hospital because it takes a little bit of time. They can be very expensive, and you want to make sure that you have the exact right virus in the hospital versus if you just go into the doctor's office with, you're stuffed up, you have a little bit of a runny nose. It could be allergies even. So they're going to just treat the symptoms. They're not going to look for the exact virus. And this gets into viral diversity. So viruses differ in their genotype. We talked about that, RNA versus DNA, their shape. So how that capsid forms, that protein outer shell, actually is one of the other ways we define viruses. And then also, is it enveloped or not? So we can use all those to break them into different families based on traits and how they actually infect us. So one example is herpes viridae, which has all the herpes viruses. This is the viruses that cause cold sores. This is the viruses that cause chickenpox. This is the viruses like EBV that cause mono. And then we have retroviridae, that's all your retroviruses. This is going to be things like HIV. There's poxviridae, which is DNA viruses that cause smallpox, monkeypox, cowpox. All of those fall into poxviridae. So we do have that taxonomic delineation for viruses still, and it's becoming more and more well defined by the genomic inputs, not just the clinical signs and is it, this genome type, this shape of capsid and enveloped or not. And this brings me to the scariest part of this lecture, which is prions, which are technically infectious and do not replicate outside of cells. So unlike viruses, which are a mix of a protein shell with DNA or RNA inside, prions are just misfolded proteins. They are what is typically normal brain proteins that misfold, and then the proteins themselves are enzymatic in nature. They cause reactions with other proteins that are the original of their shape to then turn into this other shape. Why is that bad? Well, if your proteins are misshaped, they are not doing what they should be doing. And if you have all of these proteins that are messing up all your other proteins of that type, you're going to not just not have those proteins be functional for what they're supposed to do, but also gunk up whatever system they're in. And that's why prions are always found in the brain and why they're pathogenic. Because if you gunk up your brain with a bunch of misfolded proteins that you can't clear out and just stick there, you're going to end up in this situation where you develop these neurologic symptoms and it's sadly always fatal currently. There's not a good treatment for this. In animals, it's called mad cow disease, or in deer, it's called chronic wasting disease. This is why if you go hunting and you kill a deer and you take that deer, it's suggested that you always send a sample to I believe it is the Forestry Service, and they will test it for prions for you, I believe for free, because they don't want people eating that meat, becoming infected with this, and dying of this disease. In humans, it's called Creutzfeldt-Jacobs disease, or Kuru was the original term for it. And this occurs very rarely. It's about 1 in a million people per year will develop this naturally. Luckily, we're not eating people, so it doesn't spread easily. And there's a lot that the FDA and the USDA does to make sure that we don't have animals that have these diseases enter into the food system. So, for instance, for cows, you are not allowed to sell the brain, the skull, the spinal cord of a cow. into any sort of food related system in the United States. And if that's something that's done in another country, we have embargoes. We do not import if that is going on. So we have all of these things that are in place. For instance, if a cow is found to have any sort of neurological symptoms, it has to undergo a euthanization process and then they test it to see if it has mad cow disease. And if it does, a lot more testing goes on in that herd. So there's a lot in place to prevent these things from being a widespread issue, but they still happen from time to time because they're a naturally occurring misfolded protein. So if you needed some reason to not sleep, there's your reason. And the other thing that makes them concerning from something like a biodefense point of view is they're resistant to heat, they're resistant to chemicals. So bleach isn't going to do anything to these. Autoclaves should, but sometimes they don't. There's been cases where someone's gotten brain surgery, they autoclave the surgical equipment, and then they do brain surgery on someone else. And that original person had Creutzfeldt-Jacobs disease, and then the next person gets it transferred from the medical implements that the neurosurgeon is using. And those should at least have been autoclaved. So it's pretty scary in how resistant these are. It's probably around 3 years for them to naturally decompose or longer. And there's really nothing that can be done beyond treating them very, very heavily with oxidizing chemicals that will completely disassociate any protein. And that is done in hospitals. If someone comes in with this disease, the room is treated in a way where there is no possibility of any prions remaining. It's a very thorough process, and then everything is incinerated that is touched. So, for say, if you're the brain surgeon, you're doing the surgery, they have Creutzfeldt-Jacobs disease, the smock, any eye equipment, the obviously any of the implements being used, that is all actually incinerated to prevent this from spreading. So it does fall into that viral-like acellular pathogen definition, but I want you to differentiate that from it being a obligate intercellular parasite. So it's prions are an acellular pathogen, which is like viruses, but they are not obligate incellular parasites. Um, Only viruses are obligate intracellular parasites for the purposes of this class. So hopefully you're not too freaked out. Obviously, I love talking about viruses, so if you have any questions, please feel free to ask. And I hope you will enjoy learning about viruses as much as I enjoy talking about viruses. Have a great day.